Hey guys, I'm Dr. Christopher Faraja, a doctor from Sydney, and today we're going to go through fluid homeostasis. In more detail, we're going to go through the Renin Angiotensin Aldosterone system, also known as RAAS. So, plan for today. We're going to start by having a big picture look actually at how we control fluids in our body, fluid homeostasis. We're going to have a big picture look at what are the factors at play? And there are both neural and endocrine factors. And whilst we are going to be focusing on the RAS system during this video, this first bit is just kind of like an overview, just to show you how this piece of the puzzle fits into the bigger picture. Then we're going to look at the anatomy of the juxtaglomerular apparatus. Then we're going to go into the RAS system. And I know just from the very beginning, you'll hear me say RAS system a lot, which doesn't really make sense because the S stands for system, but that's what people normally say. So I guess take it with a grain of salt. And then we're going to go into antidiuretic hormone, and then hopefully try and piece the whole video together by making a big mechanism where we look at all of it together in flowchart form to see if we can understand all the information that we've gone through up until that point. All right, so let's dig into it. We'll start with the big overview I was talking about. So, as we know, all processes within our body are controlled by the principle of homeostasis. That is, the idea that we try and maintain a constant internal environment. So if our temperature goes up, our body will try and lower it. If our blood pressure goes down, it will try to increase it. And we have homeostasis for lots and lots of processes within our body. When it comes to fluids, there's kind of two main arms. We can kind of branch it out into a neural component. We're just zoom into a neural component and an endocrine component. In other words, things that are controlled by nerves and the brain, as well as the things controlled by hormones, neuroendocrine. So in terms of the neural influences on fluid homeostasis, we have our baroreceptors. And we have both our high pressure and low pressure baroreceptors. Now, this will not be focused in today's video, but again, just to give you an idea of where this video fits into a big picture of homeostasis, which is a very complicated process within our body. And the other thing we have in the neural component is our autonomic nervous system. So our sympathetic and parasympathetic drive. All right. Now, what is the much bigger focus of today is the endocrine control of fluid homeostasis. Now, there are many. So things like ACTH and cortisol has a synergistic effect actually with the autonomic nervous system, with our serum catecholamines. Also a hormone called atrial natriuretic peptide, which we will write here as ANP for short. And then, most importantly, which is our focus for today, the RAAS system. RAAS, RAAS. And what this is short for, as we mentioned before, it's renin, angiotensin, and aldosterone. Now, this system plays hand in hand with our final hormone of antidiuretic hormone, or ADH, also known as arginine vasopressin, depending on where you look, but it's the same thing. So the focus for this video is going to be on this part here. RAAS and antidiuretic hormone, which work together quite well. All right. So in order to understand RAAS and to go through the process of each of those hormones, we do need to do a little bit of anatomy first, just so we can understand the preceding information. We can understand what's actually happening. So for those who aren't familiar with the anatomy of a kidney, the functional unit of the kidney is the nephron. And we can see a nephron on the left over here. Now, we're not going to go into detail on nephron function and anatomy, but rather big picture oversight that into the key features that will allow us to understand the RAAS system. So up the top here, we have our glomerulus. Change color. We have our glomerulus up here. Now we have our tubules, our ascending lipopendlae, or our descending and ascending limb of the lipopendlae, our distal tubule, and our collecting duct. Right? That's kind of the big picture view. But what's really important is actually if you zoom in, what's really important in the RAAS system is if you zoom into this bit in here. And if we have a closer look, it's actually what this diagram is. So this is just that zoomed in part of the glomerulus and its connection to the distal tubule, right? And we call this kind of interacting area, the juxtaglomerular apparatus. Juxta, like close, glomerular, glomerulus, okay? So if we zoom in, what we can see are a couple important parts. We can see the afferent arteriole, which is here. Yeah. We can see, well, this is the glomerulus in here. Afferent arteriole going into the glomerulus and then comes out in the efferent arteriole. Now, how do you remember which one comes first? The classic way to remember that is alphabetical order. A comes before E. So in other words, the afferent arteriole is before the efferent arteriole. Okay, so that's this bit over here, so afferent going into the glomerulus, coming out as the efferent. Now, what's important, I said, is that interaction with the distal tubule. And you can see the distal tubule here in the bit that I'm circling. What's important is this area called the macula densa. Now, the macula densa are specialized cells in the distal tubule. Or some people will say it's in the lipopendlae. It's kind of in that transition zone in a way that can detect osmolarity. Specifically, it can detect sodium and chloride concentration. And what it can do is send a local message to the juxtaglomerular cells, which are these blue ones in here. Sorry, the diagram's getting a bit messy, so let's try and clear it up a little bit. So these juxtaglomerular cells are specialized cells sitting in the afferent arteriole that can interact with the macula densa. Okay. And now the juxtaglomerular cells are what release renin. And that's why this is important. Okay. If we're going to understand how we control our fluids, we need to understand how renin is produced, the key starting player in our RAAS system. Okay. Now that we have like an understanding of anatomy, basic anatomy, let's actually look at now how this anatomy will help us to create our endocrine system. The renin, angiotensin, aldosterone system. All right, so let's start with renin. Okay. We need to understand how renin is produced in the first place. So, renin. Renin can be produced in one of three ways. It can be produced via our sympathetic nervous system. Sympathetic nervous system can act on the juxtaglomerular cells. Remember those cells near the afferent arteriole that are blue in this diagram above. When we release noradrenaline onto our beta adrenoreceptors, so the sympathetic nervous system can stimulate our beta adrenoreceptors and cause renin release. Okay. Second way is the kidney has a intrinsic autoregulation technique. What does that mean? Is that it can actually detect when we're running out of fluid, when the blood perfusing the kidneys is running out. And so just to clarify, this whole system is increased when we are dehydrated, when we're reducing our renal plasma flow. Okay? When we're running out of fluid for whatever reason, we're sweating it out, we haven't drunk, we've urinated it all out, lots of sweating, for whatever reason. That's when this system comes into place. Okay? So either our sympathetic nervous system can act on those beta adrenoreceptors on the juxtaglomerular cells to produce renin, or there's stretch receptors on the juxtaglomerular cells as well. We're going to call them JG cells for short, juxtaglomerular cells. And in this way, if we're reducing we have a dehydrated state and we have reduced perfusion to our juxtaglomerular cell because we're dehydrated, there's going to be, so if we go dehydration, it's going to cause decreased stretch, okay, which is of the juxtaglomerular cells, which in itself will cause increased renin release. Okay. And the third way we can produce renin is via the juxtaglomerular apparatus. So, how does this work? So, if we think about it, if we have a reduced circulating plasma volume, if I go up to this diagram, if we have a reduced circulating volume, then we're going to have a reduced amount of blood that can travel into the afferent arteriole, that can travel into here, and therefore go into the kidneys to form urine. Okay. So if we have less fluid going through, what's going to happen? The rate at which fluid is passing through, all the way through here, it's going to be slower, right? Decreased rate of flow. And that's simply because there's less fluid. You think about it. If you have a hose and you're pumping, 7 liters into it, it's going to shoot out really fast. But if you're only going to put 1 liter, it's going to flow a little. It's going to flow at a slower rate. Okay. There's less pressure in the system to keep it going. So if you have a decreased rate of flow, well, what's going to happen? The kidneys, the proximal tubule, the loop of Henle, all of that, it's going to start to absorb all the electrolytes, okay? Because that's what kidneys do. Kidneys, when they produce urine, they absorb the electrolytes in order to concentrate urine and therefore allow it to be excreted from the body, right? But if we have decreased rate of flow, what's going to happen? That means the kidney is going to have more time to absorb all the electrolytes. And therefore, there's actually going to be a reduced number of sodium and chloride that arrives at those macular densa cells that we mentioned. Yeah. Okay. So let's write this down just so we're on the same page. So if we're dehydrated, what's going to happen? We're going to have reduced plasma in the kidneys. Okay. What's going to happen? This is going to decrease our rate of flow through the nephron. Sorry, my handwriting is not amazing. Then what's going to happen? We're going to have increase in reabsorption of our electrolytes. Of sodium and chloride dominantly. So what's going to happen then? Our macular denser cells, remember I told you those special cells can actually detect how much sodium and chloride there are. Therefore, we're going to have decreased delivery of sodium and chloride to the macula densa. If we have decreased delivery to the macula densa, then this is when our RAAS system begins. Through local mediators such as nitric oxide, NO, and prostaglandins, PG, this causes our juxtaglomerular cells. Remember, they're right next to the macula densa. I showed you that diagram, right? The juxtaglomerular cells sitting on the afferent arteriole are right next to the macula densa because it curves around, right? Afferent arteriole over here, and the macula densa right here, right next to it. So the macula densa reduces its sodium and chloride, sends some signals to the afferent arteriole, to the juxtaglomerular cells of the afferent arteriole to say, we don't have enough fluid. We need to get more fluid. Okay? And that's when it tells the JG cells, the juxtaglomerular cells, to release renin. All right. And so now we have 3 ways to produce renin. The macula densa cells having reduced sodium and chloride. We have the juxtaglomerular cells being able to directly release renin just because detecting less stretch because there's less volume. And then also the sympathetic nervous system. Now, what does renin do? This is the beginning of our cascade. We come to this diagram here, which helps illustrate it. Renin will help to change, help to convert angiotensinogen, which is a protein made by the liver. It cleaves it off in order to turn it into angiotensin 1. Angiotensin 1, then when it travels to the lungs, to our lungs over here, our lungs produce an enzyme called ACE, or angiotensin converting enzyme. Converting enzyme. Okay, so renin's cleaved off the angiotensinogen to make angiotensin 1, goes to the lungs, has that enzyme ACE, which converts angiotensin 1 into angiotensin 2. Now, this is our key player. Angiotensin 2 can then do so many things. This diagram starts to show a couple, but let's have a bit of a deeper dive. So, what does angiotensin 2 do? The main driver of our RAAS system, which will never fail to get me the double S. But anyway. All right. What does angiotensin 2 do? 1, it causes systemic vasoconstriction. What does this mean? It tightens all the vessels in the body, particularly like our splanchnic vessels, those vessels in our gut, right, where lots of blood is kept. Causes constriction of these vessels in order to make sure that our blood can circulate around, the kidneys can get more flow, and that we're not having blood pooling in other places. Because remember, the whole idea of this system was that if you're dehydrated, you need your blood circulating. Your blood needs to go to your brain. We don't want it in your gut. So if it constricts the splanchnic vessels, those vessels in your gut, it can feed them away from the gut to more the vital organs. We need it in the heart, we need it in the brain, we need it in the lungs, right? So it causes systemic vasoconstriction. What else can it do? It can activate the sympathetic nervous system, right? Which then has its own effects that can increase heart rate and also cause vasoconstriction, right? Which achieves the same thing. All right, what else can it do? it can produce some of our key other hormones. We can stimulate aldosterone, which we'll have a deeper dive into in a second. It can also stimulate ADH. Again, we're going to have a deeper dive into what these hormones do in a second. Now, another thing it does is it causes prostaglandin release. Prostaglandin release. Now, what does this do? This is actually quite interesting. Before we go into aldosterone and ADH, let's look at that fifth point, prostaglandin release, because it actually has quite a substantial impact on our ability to maintain our fluid volume. It actually messes with the fluid composition that goes into our kidneys to make sure we don't lose too much urine. So let's have a bit of a deeper dive. All right. So prostaglandin, if we have a quick drawing of our juxtaglomerular, or like our glomerulus, okay, just a rough diagram. So this is our afferent arteriole, right? And it goes in to our glomerulus, and it comes out our efferent arteriole. I'm going to draw this so it's this again. At the beginning, the afferent and the efferent are the same size, the input and the output into the glomerulus. Now, what this prostaglandin does is it does something special. It causes preferential vasodilation of the afferent arteriole. Now, don't get confused. We caused systemic vasoconstriction, which we can't forget from earlier. So overall, everything is tightened. So there's actually less blood still going to the kidneys because the blood is being kept and saved for the brain and the lungs and the heart, and our essential organs, okay? So there is actually a reduction in renal plasma flow. I want to make that clear. But even though the total number is reduced, what prostaglandin does is it causes the afferent arteriole to dilate, but leaves the efferent the way it is. So what happens is if I redraw that afferent arteriole, now it's going to be really thick. And if I try and exaggerate it for the sake of diagram, this is our glomerulus. For the sake of explanation and clarity of understanding, this is what it looks like now. Why is this good? Well, if we have a relatively dilated afferent arteriole, this means that more, like the plasma, the circulating blood volume, will enter into the glomerulus, but struggle to leave, right? Because the rate of inflow is going to be greater than the rate of outflow. So what this does is it causes, so dilatation of afferent arteriole. And what this does is, as we said, if we have an increase in the plasma in the glomerulus but can't leave, this increases our glomerular pressure. Why is this good? Well, that means that our plasma then, because it's stuck in the glomerulus, more can be filtered out. More filtrate is actually going to be absorbed into the kidney because it's stuck there for a longer time, which means that our albumin and our proteins that are in that plasma then become more concentrated, right? Our solute can't be absorbed into the kidneys, but our solvent can. And because it's stuck there, all that plasma is actually going to be absorbed into the kidney, right? So this increases the concentration of things like albumin that then leave the efferent arteriole and go on to feed the peritubular capillaries, the blood flow to the kidneys. Why this is good is because this would increase the plasma oncotic pressure. Now, what does this mean? This means because our albumin and our proteins are so concentrated, just by simple osmosis, it pulls the fluid out of the kidneys and into the systemic circulation, right? Because these solutes are so concentrated, it drags everything out of the kidneys back into systemic circulation, and therefore we can keep it and not lose it to our urine, which is the whole goal. Remember, this happens when we're dehydrated. We don't want to lose it to urine. We want to keep it, right? So this causes fluid resorption. And that's exactly what we wanted. We don't want to lose it to our urine. The other benefit of having our increase in glomerular pressure is that because all our solvent is being absorbed into the kidneys at that time, this also decreases, right, the total, like what we call hydrostatic pressure of the efferent arterioles and therefore the blood supply to the kidneys, the peritubular capillaries. Hydrostatic pressure is the opposite to oncotic pressure. Remember, oncotic pressure from the abdomen will pull the fluid from the kidneys into systemic circulation. Hydrostatic pressure does the opposite. Hydrostatic would push, but we don't want to push from systemic into the kidneys. We don't want to lose it to the urine. So if we keep our blood in our glomerulus for a long time, we reduce the hydrostatic pressure in our systemic circulation because it's moved into the kidneys. And therefore, we have less of a push effect from systemic into kidneys. So if we look at these two processes, our systemic circulation has one, less of a push effect and more of a pull effect, i.e. both of them are going to pull fluid from the kidneys into the systemic circulation to reduce our urine and therefore maintain our fluid. I hope that made sense. All right, now let's look at aldosterone and ADH, the other two that were mentioned here. All right, now aldosterone. Let's come over here. All right. Aldosterone. So aldosterone is quite a bit simpler than angiotensin 2. Aldosterone is released from the zona glomerulosa of adrenal cortex, the endocrine organ sitting on top of the kidneys, particularly in the cortex layer. Okay. Now, what these do is they travel to the kidneys. So what we'll do is we're going to go up to our original diagram and look at the nephron. Remember that structural unit of the kidneys. So aldosterone then will come to the nephron and will go to that final bit, the collecting duct. Clear up the diagram a bit. So aldosterone will come to the collecting duct. Now, what it does is if we zoom into the collecting duct, the main cells, and from memory, I think it's like 90% of cells in the collecting duct are our principal cells. Now, oops, sorry. Computer glitch for a second. All right. So our collecting duct has principal cells, as we said. Now, if I draw a principal cell, okay, now this is the tubules on this side. So this is where our urine is. Urine. This is the principal cell, BC, and this is the blood, okay, our systemic circulation. So to clarify, this is me zooming in to a part in here, into the collecting duct, okay? Now, in the principal cells, there's the sodium potassium ATPase pump. This is a pump that causes sodium to be absorbed into the blood in exchange for potassium being pushed into the principal cell, and A+ in exchange for K+. Now, aldosterone causes an increase in the work and function of that channel. Now, what does that do? If the sodium potassium ATPase pump keeps working, then we're going to have a buildup of potassium inside the cell, which is going to create a strong concentration gradient, which will then allow potassium to leave via the potassium efflux pump over here. So, aldosterone, sodium potassium ATPase overdrive, potassium building up in the cell, creates a concentration gradient, sends it into the urine, and off it goes. Now, importantly, the opposite is also true with sodium. Because sodium is being absorbed into the blood, now there's not much sodium inside the principal cell. So what's going to happen is we're going to have an uptake of sodium moving in this way through the sodium channel. Why is this important? Simple osmosis, water follows sodium, and therefore water goes from the tubules, from the kidneys, and can then be absorbed into the cell and then therefore into the bloodstream. Why is that good? We're now no longer urinating out our fluid, we're keeping it, which again is the whole big picture purpose of the RAAS system. We're trying to maintain our fluid in times of dehydration. So that's what aldosterone does. All right. Now, the final one we said we're going to mention is antidiuretic hormone. Now, antidiuretic hormone is not technically part of RAAS, but is very much influenced by the RAAS system. Antidiuretic hormone is produced in the posterior pituitary, the posterior lobe of the pituitary gland in the brain. Now, antidiuretic hormone can be produced in a couple of ways. One, so production. One being from angiotensin 2. The angiotensin 2 can act on the posterior pituitary to cause antidiuretic hormone to be released. And we're going to mention in a second what it actually does. Another thing we can do is, another way it can be produced, is through osmoreceptors. Now, what's an osmoreceptor? These are receptors that can detect osmolality when something is concentrated or dilute. Now, these osmoreceptors, if we look at the diagram on the left, are occurring at the base of the third ventricle. So these are the osmoreceptors up here in the brain. Now, the osmoreceptors can detect when cells shrink. So if you think about it, if we're dehydrated, our extracellular volume is quite low. So all the fluid from inside the cell moves outside the cell via osmosis, and our cells shrink in size. Okay, and that's then how the osmoreceptors are stimulated. Now, there are two main osmoreceptors with very fancy names. We have the SFO for short, which stands for subfornical organ, and we have the OVLT, which stands for the organum vasculosum of the lamina terminalis. Now, I'm not going to write those out, but you have the SFO and the OVLT, subfornical organ, and the organum vasculosum of the lamina terminalis. Try and say that 20 times fast. Right, these osmoreceptors detect when the cells are shrinking, then they project to the paraventricular nucleus and the supraoptic nucleus, cause them to depolarize, which then sends down their posterior pituitary to release ADH. So, how is it produced? Antidiuretic hormone is produced via angiotensin 2 stimulating ADH release, but also there's osmoreceptors at the SFO and OVLT, which when cells shrink, project to the supraoptic nucleus and the paraventricular nucleus, which then cause ADH release. Now, what does ADH do? It goes back to the collecting ducts. And if we draw of the nephron, and if we draw another simple diagram similar to before, we have our cells there. This is our urine on this side, on the left, and our blood on the right. We want water to be absorbed, and essentially the action of antidiuretic hormone is it increases the expression of a channel on the apical side, the side of the urine, called aquaporin 2, AQP2, which allows more water to flow through this channel for water to be absorbed from the urine. So we concentrate our urine and again maintain our fluid homeostasis in times of fluid deficits. We're absorbing more water, not letting it urinate. And there we have it. Very complicated. We've covered lots of things. So let's do a very quick overall mechanism to make sure we understand. Okay? So when we're dehydrated, we have neuro and endocrine uh, influences that can help control our fluid homeostasis. Neural influences being like baroreceptors and our autonomic nervous system, which we haven't touched on today. There are other endocrine factors, things like atrial natriuretic peptide and our cortisol system, which again we haven't touched. But then there's also the RAAS system as well as ADH. So to start off our RAAS system, remember we needed renin. So we could either have our sympathetic nervous system, we could have our stretch receptors on the JG cells, or we have, remember, those macula densa, MD, macula densa, detecting low sodium and low chloride because more is being absorbed proximally in the tubule. Each of these cause an increase in renin. What does renin do? Well, renin cleaves angiotensinogen into angiotensin 1. Angiotensinogen into angiotensin 1. Now, that travels to the lungs, travels to the lungs over the arrow, with the ACE enzyme, angiotensin converting enzyme, turns into angiotensin 2. Now, angiotensin 2 does lots of things, including vasoconstriction to make sure that blood is being kept for our essential organs. It can help activate the sympathetic nervous system. It can cause release of aldosterone, which now we know causes sodium reabsorption, which allows, therefore, by osmosis, water reabsorption. And it also kicks out our potassium. Because remember, it activates that sodium potassium ATPase channel. Angiotensin 2 can also stimulate the release of ADH, which causes aquaporin 2 on the apical side of our collecting ducts to cause H2O reabsorption once again. And then we can do a quick side note. We also have our osmoreceptors, which can help stimulate antidiuretic hormone as well. And there we have it. Zoom out a little bit. That's our summary of the renin angiotensin aldosterone system, from the production of renin down to our production of aldosterone and ADH, and everything helping to save our fluid in times of dehydration. I forgot something very important in my summary. I apologize. In angiotensin 2 as well, don't forget prostaglandin release, which is a very important part, which causes an increase in glomerular pressure. Remember, and that's because it causes preferential dilatation of the afferent arteriole, which then does two things. It reduces the hydrostatic pressure of the peritubular capillaries and increases the oncotic pressure, both of which then allow decreased push from the systemic into the kidneys and increased pull from the kidneys into systemic in order to increase water reabsorption. And there we have it. We zoom out and we have a big picture look of our renin, angiotensin, aldosterone system. Hope that was helpful. Hope you learned something. And yeah, see you next video.